Hello and welcome to the Talking Precision Medicine podcast. In this series, we sit down with experts on the application of AI and big data analytics in the drug discovery space. Our guests are innovators, business decision makers, and thought leaders at the intersection of data and therapeutics. We discuss the promise, practice, challenges, and myths of AI in precision medicine. This show is brought to you by Genialis, and Raphael, its CEO, is your host. Genialis is focused on data integration and predictive modeling of disease biology to help accelerate the discovery and de-risk the development of novel therapeutics. Today our guests are Charlie Silver and Nigel Beard. Charlie is CEO and Nigel CTO of Mission Bio. Mission Bio is a single-cell analysis platform company empowering researchers to cure cancer. They are the first company to deliver simultaneous measurement of the genotype and phenotype at single-cell resolution. In this episode, we talk about the audacious mission at Mission Bio and how their groundbreaking platform enables a whole new set of questions to be answered in cancer research. We also go deep on potential clinical applications and the future of single-cell multi-omic datasets. Let's dive in.
Thank you everyone for joining us this week for another episode of Talking Precision Medicine. It's my great pleasure to be here today with two guests, Charlie Silver, who's founder and CEO of Mission Bio, and Nigel Beard, Nigel, are you the CTO now?
Recently CTO.
Recently CTO of Mission Bio. Mission Bio is a single cell platform, but I'm not going to tell you about it. Charlie and Nigel are. So Charlie, what is Mission Bio? What's your product? What's your mission?
Well, our mission is to enable our customers to cure cancer. And our customers are doing that every day with our platform, with their base at the cancer centers and drug companies, and really anybody involved in development of new therapeutics or diagnostics or basic research around oncology. That's our base. And they're doing it with our single-cell genomic platform, which does a fantastic job of measuring every molecule you would want to measure out of every single cell in a cancer biopsy.
Dig into that a little bit. So it's a genomics platform, but every molecule. So what kind of molecules are we talking about?
Well, we launched the platform with single-cell DNA. We're the only player providing single-cell DNA sequencing as a way of looking at single base resolution or gene-level resolution or copy number variation or really any scale across the genome from a DNA perspective. We were the first platform to market on single-cell DNA, and we're still the only platform providing all scales of the genome at the DNA level. It's a fully integrated platform, so we provide instrument consumable set together with the software at the back end, and the product wraps around sequencing. So we use next-gen sequencing in the middle as the readout, and the platform prepares cells for single-cell sequencing, and then we provide the software at the back end to do the full set of analytics for the customer.
There's a lot there to unpack. I guess one thing I want to try to understand, you're the only platform to offer single-cell DNA at this resolution. What's hard about DNA? I mean, single-cell RNA seems to be, cover of every science is more heterogeneity discovered by single-cell RNA. So what makes DNA hard and interesting for you guys?
DNA is interesting because it's changes at the DNA level that are ultimately driving cancer. We all know of we all know of DNA as the blueprint of life, but in fact, DNA itself is kind of an inert molecule. It's the DNA operating within a cell that actually drives cell function or drives disease. So the DNA tends to be very important for oncology. You've seen a lot of work around RNA because that tends to be a discovery analyte. There's been a lot of work around cell typing across a lot of different disease areas and in basic research, looking at how differences manifest among different cells based on expression patterns. In some sense, that's been the low-hanging fruit for the technology. It's been the easiest thing to build for the market, and that's in large part why you've seen a lot of market develop around that. DNA tends to be much harder to do at the single cell level. Turns out if you just take a cell and pop it open, the RNA is right there, but once you've done that, the DNA is still tightly packaged up in the nucleus, which kind of think of as the treasure chest of the cell. For all the reasons that nature doesn't want you to change the DNA in your cell, all of those reasons make it very difficult to access from an analytical perspective because your cell packages it up in that treasure chest. So for all of those reasons, DNA has been much harder to get to, and Mission Bio as a company, and even before Mission Bio from UCSF where we spun out, we've spent years perfecting a technology that does a very good job at DNA. So we've seen a lot of applications develop on the RNA side of the market. DNA tends to be very important for oncology research, and especially for delivering meaningful discoveries into the clinic. And that's why we decided to focus on DNA, and been sort of a sweet spot for us that's made it much harder for the others to get into.
Very interesting. Can you talk a little bit about some of those applications, maybe a couple of your early success stories where you and your customers, your partners have discovered something interesting?
A lot of the early work on the platform was built off leukemias, and that's because we launched the platform with some standard panels for leukemia. It's also because that's naturally the application sweet spot for single-cell DNA, because the genetics tend to be very well understood. There's been decades of work understanding the genomics of leukemia, and the genetic landscape has been reasonably well mapped out to the point that you know what to do with single cell. And that's why a lot of the early work on our platform has focused on the leukemias, although we've also built out a pretty substantial application set across solid tissues and many other disease areas as well. Some of the early work on the platform was published from some of our customers at the cancer centers. There was one study in particular that was done by a collaboration with UCSF and UPenn, and those researchers looked at therapeutic resistance to targeted therapies for leukemia. And they found, using single-cell DNA, that with the sensitivity that single-cell gives you, together with the mutational profiling of single-cell DNA, they're able to map clonal architecture to the sensitivity level that gives them the ability to identify resistance mutations and resistant clones that they're able to track through treatment and understand the resistant mechanisms. Single cell gives you another order of magnitude on sensitivity on top of bulk sequencing. So bulk sequencing conventionally gives out at about 5% variant allele frequency. If you're really good at it for some clinical assays, you're able to get down to 2%, but typically not lower than that. Single cell with Mission Bio's platform gets down to 0.1% sensitivity. And that order of magnitude can become very important for studying the rare clones that are going to become resistant to therapy, because they exist at populations well below 1%. So using the single cell platform, our customers at UCSF and Penn published a study showing resistant mechanisms to gilteritinib, which is a targeted agent that's increasingly used to treat acute myeloid leukemia, and identified resistance pathways based on the Tapestry platform.
Wow, that sounds very important and almost kind of immediately clinically actionable discovery. That's neat. I can imagine similar use cases for things like minimum residual disease and potentially even early detection, given those sensitivity levels. That seems like any number of potential applications there. Recently, you guys had a pretty big announcement, though, that your platform's not limited to DNA anymore. Tell us a little bit more about what else you're analyzing.
Well, our goal as a company is to provide the oncology community with the complete tool set that's needed for single cell to carry you from discovery all the way through to the clinic. And recently, we expanded the platform to include DNA and protein simultaneously at the single cell level. And this is the first time that the space has ever seen the ability to genotype and phenotype cells at the single cell level, which is the resolution of cancer. Every cancer cell is different to some degree, and that difference manifests at the single cell level. So that's where the resolution matters for the study of cancer. And now for the first time, our customers are able to basically bookend the dogma of biology by measuring the DNA and then the functional result of the DNA at the level of the cell. And that opens up a whole host of really important applications for cancer discovery and biomarkers and delivery for different disease areas within cancer.
And so how does that work, both from the DNA and protein side? Are we talking about whole genome, whole proteome, or are these going to be panels? And then how do you design the panels? Are they specific for diseases or particular known markers?
On the DNA side, it's the same product we've been selling. So you're able to run any type of DNA measurement from single base through to gene level, through to copy number, or tiling up across the genome. So it's exactly the same product. On the protein side, you're running antibody panels, and very similar to the way flow antibodies run, you have an antibody that instead of being conjugated to a fluorophore like you would do for flow, you take exactly the same same antibody and conjugate it to a DNA barcode. And then exactly the same way you would do for flow, you stain and wash the cells at the front end, and that DNA barcode is now bound to the surface of the cell. And then the cell runs through the platform, really identical workflow. And by the time you get to the sequencer, you're reading out single cell DNA with a simultaneous measurement of protein expression on every single cell. And really a lot of advantages to that. Multi-omics is becoming increasingly important across oncology, but especially for single cell, because there's an understanding across the space that for biomarkers or for basic discovery, if you're just looking at one analyte, you're missing the picture. And that's where, resistance to therapy tends to land. It's somewhere in between all of the analytes stacked together. Right. So now that you're able to look at DNA and protein together out of the same cell, you have just a much finer lens and much better resolution into the underlying biology, and really just opens up applications across the space.
What's making my antenna tingle here is, so my company spends a lot of time talking to immuno-oncology therapy companies, so pharma companies developing immunotherapeutics of all kinds. And, protein is such an important analyte for a lot of these applications. Whether you're doing a cell therapy and you want to understand, how much of the target is there, how much of your therapy is there, whether you're looking at antibody therapies or oligotherapies, what have you. The proteome of the immune system is in some ways a lot more straightforward to measure and understand than say RNA, where you have all sorts of pathways going up and down and so forth. So it seems like the application space just in immuno-oncology alone, there's just, treasure troves here for people to think about how to develop.
Yeah, that's the way to think of it. DNA on its own is the blueprint of disease, the same way it's the blueprint of health. And then protein is typically the way we interact with disease. So, you try to modify proteins or in some way change pathways based on proteins in order to kill the disease. And once you link the two together, you finally have that interplay between them at the single cell level. And, it's something that we've been doing, or the space has been doing for a long time at the bulk level, and then, either making inferences down to single cells or else applying some complex algorithm to be able to link the two. But fundamentally, you'd like to be able to integrate them at the single cell level because that's where it matters. So we're really providing the space for the first time, the ability to do true genotype and phenotype of single cells, so you can, really in an integrated way link the DNA mutations that are driving the disease together with the proteins on the surface that ultimately you're using to treat the disease or to diagnose it.
And so that DNA barcode that's conjugated to the antibody, that has a GPS coordinate that lets you know which cell that protein's coming from. Is that correct?
It's got a barcode identifying the cell the same way you've attached on the DNA side. So it's the same barcode that reads out. Exactly. They're physically linked at the single cell level.
That's brilliant. I'm also thinking about like neoantigen development. Man, the number of things you could do with this are just astronomical.
Neoantigen discovery has been a really hot application space for us. Basic biomarker discovery is also very important for a lot of disease areas. You know that there are rare stem cells driving the disease, and you can typically identify those stem cells based on their genetics, but you don't have phenotypic biomarkers for them. And they're in such a rare cell population that bulk sequencing doesn't help you. And without any phenotypic markers, there's no flow sort that's going to identify them. So this is a way of getting down to those very rare cells based on the genetics. And then once you've got the genetics, identify the protein biomarkers that can then be used from there. Another really interesting application that's gotten a lot of attention since we launched has been CAR Ts and cell therapy in general. And to me, this is one of the hottest areas of single cell, and I think one of the most important applications for what we've been doing to enable the entire cell therapy industry. There's a whole collection of assays that are built out to characterize cell therapy product, from flow to exome sequencing, digital PCR, and in some cases they're using single cell RNA, really everything in between, and then stitching it all together into one data package that characterizes the cell therapy product. Now that we're able to look at copy number variation, at a single fold change level, which is what the FDA tends to use to regulate based on or to identify dose toxicity for every drug. now we can identify copy number at the single cell level, which is really where it matters, because that's, fundamentally it's a single cell product, but then link it together with the phenotypic result on the surface of the cell, again, at the single cell level. So you're integrating assays and providing just a much cleaner data set.
That seems like a really important area going forward, especially as we start thinking about how to democratize access to cell therapies and thinking about building more off-the-shelf products in that regard, or quickening and making it much less expensive and less error-prone, the autologous therapies. I think assays like
that's exactly right. And it's really all of the above. It's the cost of integrating all of these assays with the bioinformatics to glue it together. And that cost is not trivial, but we pay it because these cell therapies are really important, but also the time to answer. Because running all of these assays stacks up into days and weeks that can be short-circuited with a clean single-cell readout.
I think part of the theme of this podcast is to always kind of bring the conversation back around to data. And one of the amusing bits is that very often when you get a bunch of biologists talking about data, we're used to kind of bench assays. And so as soon as you start talking about sequencing, it feels like big data. But bulk sequencing never really got us there. With single cell, we actually start to approach fairly large data matrices. And now you're talking about single cell for different analytes. So, Mission Bio and your platforms are going to generate a lot of data for a lot of people. How do you see that data shaping your identity? Do you think it's going to be one of those things where customers just look at you guys as a way to get more and more data? Or do you plan to bring some of that sort of data molding and shaping in-house and help show people what they can do with it?
People tend to think of single cell as complex data sets, but in reality, when it comes down to the work the customers are doing on the platform, it's actually simplifying the data. And by simplifying it, you get more meaningful insights out of it. And what I mean by that is this: a lot of times what I hear back from clinicians who are running the platform to study clonality of cancer biopsies is that when they're running bulk NGS, again, to get down to that 2 to 5% variant allele frequency, they're looking at a bunch of variants on a readout, which could be several pages long, and it just tells you that you've got this variant in this fraction of the DNA population, and it's up to you to make that interpretation of what to do with all of it. And it could be several pages worth of different variants, and it's up to you to interpret it. What the clinicians tell us is that single cell is basically turning all of those variants into something that's more meaningful for them, because what you really care about is the clones. It's the collection of cells that have each combination of variant. And once you're looking at the combinations of variants in different cell populations, you have a deeper level of insight into the biopsy, and you have a much better way of understanding what to do with it. So when these clinicians are looking at page after page of an NGS readout, what they're really reconstructing in their head is, okay, what are the different clonal populations? And based on that, what are, possible treatments for the patient, or, what's the possible evolution of this biopsy? Single cell fundamentally simplifies that. So you're looking at clones instead of a giant collection of variants, and you have much better confidence in the insight that you get from that. So I just want to make the point that single cell in some ways is actually simplifying the picture for them, even though you tend to think of it as more complicated data. It is true, though, that our data sets are becoming increasingly more complicated, and there's just a much richer level of insight and information that you get out of the sample. Once, at every cell that matters, you now have every molecule that cell has to offer you. So a really important part of our solution is the software at the back end that turns that into something meaningful for the customer.
So in addition to having your R&D wet lab and some manufacturing, you've also got bioinformatics and software, and obviously with Nigel on board, a data science team. Tell me a little bit about, I guess, the challenge of running such a multi-headed hydra, but also how you think about those teams kind of integrating, and maybe you can then toss it over to Nigel to comment a bit on his vision for his side of the show.
You've hit on an important point. It's a challenge, but I think it's also one of the most important points, and really one of the most rewarding points about being in a business like this. We sell a fully integrated platform that's really built from the bottom up, and it's got everything in it, from hardware to microfluidics to surfactant chemistry to biochemistry. molecular biology, software, and really everything in between. It's a very complicated but fully integrated system, and it takes all different talent working together to be able to pull together a system like that. The defining features of our culture are that we are curious, collaborative, and driven by our mission of enabling our customers to cure cancer. And those three qualities of our culture were very thoughtfully built in as a way to enable this highly multidisciplinary team to trust each other and to question each other, but also be very respectful and openly collaborative with each other, because everybody at Mission Bio is the very best at what they do. But by working together, we produce something that's much better than any of the pieces. So it's really important to foster that kind of open and collaborative environment. And I think having that type of environment really has enabled us to build a very, very complex product in a way that simplifies the technology and simplifies the output for the customers. And I'll turn it over to Nigel to talk some more about how the teams come together around that.
Yeah, Nigel, who do you like to go in and challenge on Monday morning?
That depends on the sequencing readout we get on a Friday. But I think, as Charlie pointed out, this is a systems approach. I am not a biologist by training. I'm kind of a biologist by osmosis. I'm a technologist. I love technology. I love providing platforms and systems that provide researchers and clinicians a way to probe the insights that they've set up during their careers. I think we're at a defining moment, I think, right now with single cell in particular, that we're able to ask questions of a system, truly multi-omic questions, and see what's going on in that. We do have a challenge in sort of simplifying the data set out so it's easily readable. But the actual data sets, the information that they contain, are very complex, and they match the complexity of the biology that's going on now. So we have the ability to probe it and ask pretty deep questions. So this team is a multidisciplinary team. It is chemists, biology, bioinformaticians, which they've only been born over the last 10 years through the next generation sequencing. I was on the precipice of that. Do we train biologists to learn computational methods, or do we train computational people to learn biology? This born the bioinformatician, and now you've just touched on it. We're about to really hit big data in the life sciences for the first time. And that's a really exciting data point. But we have a multidisciplinary team that come together and think through experiments as a system rather than just from a chemistry or biology or a data point of view.
Well, let's stay here for a second. When you say we're about to hit big data, I want to come back to your training and background, which has taken detours outside of biology to areas with maybe more data. But what do you see in biology as being big data? What would cross that threshold in your mind?
Yeah, I think I'd have to go back to a point in my career, where I started on the next generation sequencing way. And this was just amazing. Post 2001, where we sequenced the first genome, this was Celera and Applied Biosystems. I joined those companies. And this was an exciting moment. We had this full reference genome, been done for the first time. We spent millions of dollars on this. It took years to complete, but we had this very rich data set. And then next generation sequencing was born because we had a reference. We could then say, okay, we could do this quicker because we know what the answer should be, and we need to chop up smaller pieces, do it faster, better, cheaper. And that spawned a whole bunch of new technology. And I was on the 100 genome project, the 1,000 genome project, even the 100,000 genome project. And I was so excited. I was carrying hard drives around from building to building with terabytes of data. And I thought I was in big data. I thought this was the moment. And then when it came to the primary, secondary analysis that we started to go through, the finance industry, for example, has been bidding big data for years. They've been leading the trend. They can now predict, what the market's going to do with nanoseconds of accuracy, and they trigger sales. None of us are going to be able to actuate these, but computers are already out there using predictive algorithms to take action based on, big data. And when we were doing this kind of analysis, looking across, I was in the HID, human identity, looking for commonalities in SNPs or SDRs and working with the FBI. Could we use this data in some way to help some difficult problems? We've kept hitting the wall. There wasn't enough data. It sounds crazy, but these are large, very deep, rich data sets, but you need more. You need more temporal points within this data. And single cell came along, and now we're generating, tens of millions of single cell genomes every single month. And these are also time-based. We have pre-treatment, post-treatment, during treatment, microneedle aspirates. You're able to get every sample you take is 10,000 samples in itself. And then you've got millions of data points within those. So now we're generating billions of data points. And this gives you, access to really do some interesting and insightful things that we've never been able to do in the genomic space.
So, Charlie talked about a bunch of these, but I'd love to get your take. It seems to me one of the real kind of 0 to 1 impacts that Mission Bio and perhaps some of the other single cell platforms will have is there are now a whole suite of biological questions we can actually ask. Before we could wonder about them, but we couldn't ask them with the hope of answering them. What do you see as some of maybe the cooler, say, killer applications that you'd like to apply Tapestry and Mission Bio's platform to?
Yeah, so I think, again, not a biologist, and this is about us providing a technology and platform to those wonderful thought leaders out there that have, to some extent, I see it similar to astrophysics. You've got your theoretical astrophysics, and then you've got the experimental ones that can actually now have platforms that can test those hypotheses. I believe that we're about to supply the Hubble Space Telescope, if you like, for the genome world. We're going to be able to give them that depth of insight into connected analytes. So this is, unlike a company like yourself where, we want to interdigitate all of these disparate layers, protein, RNA, DNA, because the cell is a single complex unit. And you want to be able to interdigitate that and see how they communicate it. Mm-hmm. And we're going to be able to generate that from that one single data set. So what will they be able to do? Like Charlie said, the DNA is very important. That's a blueprint of life. We've got, the next layer, which is the RNA, and we've also got the protein. So you've got a perturbable system that you can monitor, supplying something into that cell, what happened within that cell, and what was the result of what happened during that mechanism. And this is just a phenomenal platform that's enabling, researchers and then eventually the clinicians to be able to take advantage of that.
No, that makes a lot of sense. I love the Hubble telescope analogy. Let me ask maybe a similar question, but more of a technologist question. So one of the kind of worlds that I'm steeped in now is the world of artificial intelligence as applied to healthcare. And I spend a lot of time on various advocacy groups and just kind of thinking about that. It seems to me that one thing that single cell has done is given computationalists in the biology world enough data to actually start playing with some of these cool AI algorithms. And so whether it's for imputation or autoencoders to try to build a low latent space representation of RNA or whatever. What are some of the AI tools or the algorithmic tools that you're excited to start playing with now that you've got these data sets?
Yeah, I think, let's be honest around the table here. AI is not new. It's been around for many, many years. And I think, we've seen uses of that in many different industries. And, I tend to see it as augmented intelligence rather than anything artificial. I think it's just a fundamental thing for me that this is not about replacing jobs. It's about doing things that, a human would just tear their hair out trying to do or probably couldn't do. Right. And so, just setting the scene there that this is not about replacing, smart individuals to be able to do some of these things, but being able to look at 4-dimensional data sets that have billions of different combinations and be able to see patterns within that and infer future predictive patterns from that. Mm-hmm. That is what, AI, in particular machine learning. And then the next level of machine learning, which I'm really excited about, is these neural networks and deep learning, or the ability to take massive amounts of data that are known. I think very important here, one of the things I learned is, it is garbage in, garbage out. So you have to make sure, I think the first year at Mission Bio, that's my only year so far, has been making sure that our data sets are in clean format. We need to be able to train correctly. We need to be able to parse our data sets, that we have training sets, learning sets, and then probing set. Yeah. And I think, for me, most importantly this year has been about our data infrastructure as a company, setting that up, making sure that we're able to take in that data and parse it in particular ways and keep it in certain formats so that we're able to trust, the kind of information that we get out. But we have really, now started to spark the minds of the Googles and the Amazon, the cloud-based infrastructures that are letting us store the data there. They obviously see us as the Netflix of the biotech world. This amazing amount of data, but could we apply these novel machine learning, deep learning algorithms to better not only the products that we offer, but also insights into the biology?
No, I'm sympathetic to the challenge of the data infrastructure, data cleanup part of the equation. It doesn't sing, but it's really important to do at the outset. It occurs to me, though, that you alluded to this idea of coming up with kind of predictive patterns because you are already measuring genotype and phenotype. And as you presumably add more analytes throughout the kind of central dogma and the less central dogma to your stack, you might have some really cool predictive power. Like, based on the genotype, can you nail what the proteome is going to look like around these cells? Or maybe you want to do it in the other way because it's easier to measure the proteome or what have you. But
you hit on a very important thing about, these neural networks and how you see the outputs of these systems. They are essentially a huge biological probative data set that you can go in and ask questions forwards, backwards, from the inside out, ways in which you would, have to do in a wet lab, millions of experiments to get these through. And I think for me, the predictive nature of what we have here, we have a very good example. And I think, Charlie gave me the permission to do for the first year is to sequence every single custom panel we ever make. This is the amplicons that you would choose for a specific disease that you have interest in. And we sequenced every single one. And we have a minimum bar by which we want the sequence in panel performance to achieve. It has to be 90% or above. If it dropped below that, we would tweak from equimolar to, change the molarity of the high flyers or the low flyers. Human nature is to say we're just going to double it or we're going to half it, and then we see. With an AI where it doesn't understand, whether half or double is meaningful, it's actually going to use finite quantities and spit that out and give us a program for a robot to actually tweak the panel based on prior performance. So in the future, not too distant future, you'll come to our site and you'll be given a panel performance predictive score based on all the panels prior to it. That amplicon you choose, in particular, if it's in a different panel, it may respond differently. And so we'll be able to use all of that data that come before to design better panels from the get-go. And that's just one example where we're taking other data that we have generated with other customers, and you would get the benefit of that as a mixed mode mission bias.
Right. So you've kind of outlined one use case for this advanced analytics, which is product improvement and making sort of your platform bulletproof for users. You'll probably, however, given the data sets you're accumulating, also be kind of the keeper of the keys to how you think about extracting biological insights. Do you envision having sort of a data science product or partnership program so you can help your partners, these thought leaders in cancer, do more with their data?
Yeah, I certainly hope so. I think, it's clear to delineate that the metadata that we hold is no personal medical information. We are just getting the generation sequencing output. We do have partners where we are sharing that data together and being able to look across multiple different PIs that have done multiple sets of experiments, not together, but are looking at, similar inferences in certain cancers. And they have the metadata on the patient side. So we can see, did this patient with this type of biomarker respond well for treatment in this particular case? And what would happen if we had a different patient from a different set? Would they respond equally as well? I think that is where we're going to start to see the power of that. But, this is, for me, is a journey. I think anyone who's been involved in big data, this is not something that we turn around and say, hey, we've got tens of millions of single cell genomes now, and we're going to be able to predict how, cancer work in the body. But I think one of the most exciting things for me is we're going to move away from this sort of bulk preventative treatment mechanism to more of a precision-based, unique single cell approach for individuals. And that's really going to start to allude to, solutions that I can't even imagine right now. But it's very exciting to see our customers, see some of the insights that they're getting from the data sets very early on.
Tell us a little bit about, I mentioned you took a detour out of science. So you did grow up with a science and engineering background and worked in biotech. You described being on some of the early next-gen platforms. I remember the very last experiment of my thesis work was a 454 experiment. Yeah, so I remember those heady days. But then you moved into the consumer space. What was that about? What was that like?
Yeah, that was kind of a moment in life where I had mentioned getting involved in this next generation sequencing wave. And I started to lead the team at Life Technologies in the HID department. And this was really now taking this next generation sequencing platform and how could we apply this in the world of CSI, working with the FBI, looking at STRs not only, but then moving into SNPs. Could we build a molecular witness using SNP technology? Could we, get facial morphology, hair color, age, etc.? And Life Technology at the time just got acquired by Thermo. And I believe that the big data is going to arrive in the DNA space, but I'm not going to have a playground to do that if I'm in the life sciences. We're kind of the laggards. We need to be the laggards. There's a lot more oversight in what we can do in this area. So is there a space which I could go learn more in person about big data, AI, machine learning, deep neural networks, work with Google, IBM Watson, even one of my heroes, Wolfram Alpha, worked on a sort of machine vision algorithm. And so that's where I took a a bit of a leap into the consumer space there for, about 4 or 5 years to be able to play around in big data and not have the impact of what would be life and death.
What did you learn from the big data there? What did you learn was big data? What did you learn were the limitations or maybe the possibilities?
Yeah, limitations and possibilities. First is just the amount, starting at ground zero and having no data and then having to collect it. It took us 2 years to collect meaningful data. And, I'm going to tell your listeners now, I went to the fashion side of the world. This was in shoes, working with Nike and Adidas, and, thinking how could we make really cool footwear not only look good, but also fit the form and function that you were going to do, whatever sport it was you were going to do in that. And that involved taking photogrammetry data from your foot, creating 2 to 3 million data points from each of those feet, putting it into a manufacturing environment that could augment a consumer product based on that data. I initially thought, hey, we could get, 10,000, 20,000 people and scan those and get enough data points so that we could do something meaningful. And I actually took a partnership with some of the, the leading shops in the malls to be able to generate reams and reams of scans and data to make it actually meaningful so that we could actually do something with that product. And we used 3D printing, which was a mechanism which would take the 3D data and augment product. And so we had to build everything from scratch, really, to be able to augment. But once we did that, we started to see, true, real, meaningful movements and moments in the algorithms with which we used over the top of this big data to predict fit based on 3 photos from a smartphone, which was kind of cool.
So it sounds like 2020 is going to be the year that Mission Bio really steps into its place as the big data company in the single cell space. Charlie, so we're recording this just on the eve of New Year's Eve. I've been doing a lot of thinking about sort of what Genialis has done in 2019 and getting ready to tell our story to the world about what we're planning to do in 2020. What's Mission Bio's version of that? Looking back, what are you proudest of in 2019? What maybe are you surprised that you got done that was a stretch goal?
I think in 2019, I'm proudest of what the customers have done on top of the platform. This is a business. We have some of the brightest minds out there in engineering and product development and basic science that goes into platforms. But ultimately, our customers are the heroes because they're the ones who are out there on the front lines of cancer every day, studying and coming up with better treatments for cancer. And I'm really proud that we're now baked into all the leading cancer centers in the U.S. We're adopted by a lot of the leading pharma companies to help accelerate their drug pipelines. And I'm really just inspired by the work that our customers have done on the platform this year. Heading into 2020, I think the last couple of years have been the leading edge of the wave in single cell. I think we've seen the research side of the market develop in a big way to the point where it's no longer cutting edge, it's mainstream, and you really have to be able to use single cell to enable your discoveries and to enable the best research out there. So I think we've reached the point where single cell is mainstream. I think we're now at the leading edge of the wave of multi-omics. And to me, I'm just really proud that we brought out a platform that's for the first time ever able to bookend the dogma of biology, give the customer every analyte that they need to be able to measure out of the cell. And I think next year really is going to be the year of single cell multi-omics.
I'm looking forward to it. I hope that we get to get our hands on that data as well. So for new customers, pharma companies or cancer centers that haven't adopted Mission Bio yet, how do they get started with you?
Well, we sell direct here in the U.S. and we are selling worldwide. So it's mostly a matter of talking to some of the folks who are already using the platform because they're all around you, understanding how they're able to use it, looking at the publications that are out there and more coming online to get a sense of what single cell can do for your research. And then really, we're the platform enabler. We provide both custom panels and catalog targeted panels. But we can also build in any content that you, the customer, have in mind for your application. So it's really the customers who are building applications on top of the platform. And it's a matter of working with us to understand what's the right content for your specific application.
Very cool. I hope some of our listeners give you guys a call and get started right away. Thank you both, Charlie and Nigel, for joining us on this episode. I learned a lot. Certainly Mission Bio is way more than just DNA sequencing. This genotype phenotype technology, and as you stretch the central dogma, I think it's going to be earth moving. It really will.
That's awesome. Thanks a lot, Ray.
For having us.
This has been episode 17 of Talking Precision Medicine. To learn more, feel free to reach out directly to Mission Bio or to Genialis. Both companies will be out and about at JPM and happy to connect there or anytime after the circus leaves town. Also, please share the episode with your colleagues, leave a comment or a review, and stay tuned for the next one. Thanks for joining the conversation.